This is a DNA strand. A DNA is double-stranded with a double helix, with each strand made up of sugar phosphate. Phosphate is this, and sugar is the two connecting these. This is the backbone, it's made of touch bases. Adenine, guanine, cytosine, and thymine. Aryl deoxyriboic sugar and phosphate molecule. One of the four nitrogen bases of these is called a nucleotide. Along the molecule, there are long sequences of genetic code made up of bases. The replication of these long sequences accounts for the replication of the genetic code of the organism. For example, the adenine we paired up with thymine, and cytosine we paired up with guanine. This makes up a DNA double-stranded, and it's the first part of DNA replication. The first step of DNA replication is to unzip the DNA. This is carried out by an enzyme called a helicase, which breaks the hydrogen bonds holding the complementary bases of DNA together. So DNA replication is when the DNA is unzipped. The separation of two single strands of DNA creates a Y shape towards the replication fork, which is at this end, which is where it will be called the leading strand, which is the left side of the DNA. The other strand is oriented at 5 to 3 direction, which is called the lagging strand, which is this. This is called the leading strand. These two single separate strands are put on each of these bonds corresponding to match up two single strands which acts as a template of DNA. Two separate strands will act as a template for making new strands of DNA. As these two strands are oriented differently, two strands will replicate differently, going either way, one going up and one coming back down again. DNA replication occurs in the nucleus, where the polymers are these are enzymes that create DNA molecules by assembling nucleotides, which are the bases, to the building blocks of DNA. Once the polymers have made the DNA nucleotides, they are added to the DNA parent cell with two single strands, which are added to the parent strand. One's called the lagging strand, which is this one. One's called the leading strand, and that's the lagging strand. That's 5'3", and that's 3'5", coming back the other way. This is the final product of DNA replication, where the helicase is in the middle, which is the purple thing, which is this. And these are the two complementary single strands joined together. To this assignment is once the helicase has unzipped the DNA in DNA replication. Next bit is transcription and translation, where transcription is a process by which DNA is copied to messenger RNA, mRNA, which carries the information needed for protein synthesis. Transcription takes place in two broad steps. First, a pre-messenger RNA is formed with involvement RNA polymerase, polymerase enzymes. Which happens over here. Where the polymerase connects the bases together to another DNA strand, where this is made up of phosphate sugar. The process lies on Watson-Crick base pairing, and the reluctant single strand of RNA is a reverse complement of the original DNA sequence, which you can see here and here, single strand. The pre-messenger RNA is then edited to produce the desired mRNA molecule, which is a process called RNA splicing. The strand that contains the gene is called the sense strand, which is this, while the complementary strand is the outside strand, which is the non-coding strand. Pre-RNA is the mRNA is produced in transcription, which is a copy of the sense strand, which is this, whereas the anti-strand that is transcripted. With this, the messenger RNA is connected to the anti-strand. The blue base here is uricase, or uricase, which is swapped with thymine. Transcription takes place in the nucleus. The strand opposite messenger RNA is called the coding strand, where the messenger RNA is copied. After transcription is translation. Translation is the process by which a nucleotide sequence of mRNA is converted into amino acid sequence of a polypeptide cytoplasm. The mRNA formed in transcription is transported out of the nucleus into the cytoplasm to the ribosome, which is this. The first stage of translation is initiation. Transfer RNA, or in other words, tRNA, is a single strand of 80 ribonucleotides. It functions as an interpreter between nucleic acid and peptide sequences by picking up amino acid and matching them to the proper codons in mRNA, which is the bases. Here it directs protein synthesis, messenger RNA, mRNA, where it's not directly involved in the protein synthesis. It transfers RNA and RNA, which is tRNA, which is required for this to happen. The process by which mRNA directs protein synthesis is the assistance of tRNA, which is called translation. An anticodon, which is the other three bases, on tRNA, is a complementary to the RNA codon. An anticodon can recognize and bind its complementary mRNA codons to the others. At the three end of the tRNA strand is where the amino acid attaches to the tRNA molecule, which is shown right here. The ribosome is a very large complex of RNA and protein molecules. Each three bases stretch of the mRNA triplet is known as a codon, and one codon contains the information of a specific amino acid. The ribosome consists of a P side, which is peptidal, and an A side, amino side. This moves along the mRNA, as shown here. The first mRNA codon is UAA, which is uracil, adine, and tRNA for anticodon is AUU, which is adine, uracil. This attached to a tRNA is a corresponding amino acid, methionine, which attaches to three which attaches to 3 codons opposite it. These that attach to the tRNA are called anticodons. As the ribosome moves along the mRNA strand, another tRNA comes in with the next amino acid attached to it. The next 3 codons, anticodons, are corresponding to the mRNA codons, which in this sequence is cytosine matched with guanine, guanine attached to cytosine, and adenine attached to uracil, where this will attach to twins, and the next amino acid is attached, which will cause a chain, which causes another amino acid chain. Once that is been passed, this enters the A side of the ribosome. Once that is done, the next amino acid comes in through the A side of the ribosome, comes through the A side of the ribosome as the ribosome moves along. The next tRNA comes through to the A side of the ribosome. The next amino acid enters the A side where the third mRNA codon is GC, which is guanine, and cytosine, and tRNA anticodon is cytosine, guanine, where these will attach. The next amino acid will attach to the chain for the other amino acids. This stage is called elongation. When the stop codon comes into the A side of the ribosome, an enzyme called release factor, which is this comes into the A side of the ribosome, and translation is terminated. This is the third stage of translation. This is when the newly formed protein is released. That is called protein synthesis. The release factor also breaks the bond between the tRNA and the polypeptide. The polypeptide and the tRNA are released. The released polypeptide forms a protein, which is formed by the amino acids. When the amino acids join together, they are joined by a peptide bond. The second amino acid is joined by a third amino acid, which is another by a peptide bond. When the amino acids have joined, at the end they are called a protein. The peptide bond is formed between the new amino acid in the A site and the green peptide chain. The tRNA is released from the P site and the ribosome advances one codon in the mRNA strand. The names of the amino acids are red being methionine, light blue which is the second amino acid, is called arginine or arginine. Next amino acid, which is green, is called a proline. Fourth amino acid that is attached to the amino acid bonds, which is connected by a peptide bond, is isoleucine. The fifth, which is dark blue, fifth amino acid, which is another attached by a peptide bond, which is glutamine. And the last, yellow, of the amino acid of the protein chain is called valine.